Okay, so our topic today is cell cycle and cell division. So the objectives of the today's lecture is to describe mitosis and meiosis, stating their importance, and to be able to state the phases of the cell cycle. So first, what's the purpose of cell division? Why do we have cell division? For growth, we need cell division, right? So for a small baby to grow into a complete adult, we need cells to grow, right? Then for cell replacement. So for cell replacement, so if we, for example, take the stomach, uh, there are cells on the stomach surface, right? So the topmost cells usually break off with time. So you need to replace those cells. So that occurs by cell division. And for cell regeneration, so if you develop a wound, or like if you remove a part of your liver, it regenerates new tissue to replace the lost tissue. So, and for sexual reproduction, to produce a new organism, you need cell division. So chromosomes. So when you are talking about chromosomes, there are some words you have to understand clearly. Those are chromosomes, chromatin, and chromatids. So if you look at the cell, you have a nucleus, right? This is the nucleus. So the nucleus is the control center of the cell. It also stores genetic material. So in this nucleus, you have genetic material as chromosomes, like this. Right? So you have 46 chromosomes like this. These are known as 23 pairs, 23 maternal chromosomes and 23 paternal chromosomes. That is 23 you receive from the mother, the maternal ones, and 23 you receive from the father. So, each of these 23 maternal chromosomes have a similar looking paternal chromosome, which is the same shape, but the genetic material may differ. So when you take a resting cell, these chromosomes are uncoiled. The DNA strands are uncoiled. So you can't see the proper chromosome like this. So this uncoiled genetic material is called chromatin. Chromosomes are DNA strands which are coiled and arranged like this. So during cell division, chromosome replicates itself like this. So you have two similar strands. So this whole thing is called a chromosome, and this whole thing is also called a chromosome. And these separate strands, the equal looking strands, are called chromatids. So this chromatids of the same chromosome are called sister chromatids. And another thing, so you have a paternal chromosome and a similar maternal chromosome. So these similar chromosomes are called homologous chromosomes. So these homologous chromosomes, so if you take one locus, that is one site, they both code for the same gene. So the genetic material may differ and may give different results, but they code for the same thing. Other thing you have to know is, so you have two pairs of DNA, two pairs of chromosomes. So as you have two pairs, they are called diploid cells. So if you have only one pair, as you will see, which occurs in the formation of gametes, if you have one pair, it's called haploid. Right? So yeah, I think that's the basics. You can see a chromosome here. This is a duplicated chromosome which have two sister chromatids, right? So you can see how the DNA is coiled and arranged into this chromosome. So here you have a chromosome, and here you have a duplicated chromosome with the two sister chromatids. So you have a part called the centromere. So during duplication, the two strands are attached together by the centromere. So centromere is the genetic material. Around this, there is a protein structure. This protein structure is called a kinetome. So then you move on to the cell cycle. If you look at the cell cycle, there are two main phases: interphase and mitosis. So this is the interphase. It is much longer, and the interphase also can be subdivided into three parts: gap 1 phase or growth 1 phase, synthesis phase, and gap or growth second phase. Right. So in the gap 1 phase, in the cell, the organelles divide and the cytoplasm expands. So in the synthesis phase, what occurs is the genetic material duplicates. So the single-stranded chromosomes become sister chromatids. Like this, this occurs during the synthesis phase. And in growth 2 phase, there is further enlargement of the cytoplasm and growth. And it prepares for the mitosis. Now, mitosis is divided into four parts: prophase, metaphase, anaphase, and telophase. And at the end of the telophase, we have cytokinesis. That is the division of the cells. So during this interphase, in the nucleus, the genetic material is not organized as chromosomes. You can see it as chromatin. Okay. So here you can see the cell cycle. There is gap 1 phase, synthesis phase, gap 2 phase, and the mitosis with prophase, metaphase, anaphase, and telophase, then cytokinesis. So at the end of the cytokinesis, cells can again enter the G1 phase, but some cells stop their growth. Their growth is arrested. So, for example, nerve cells. Nerve cells don't divide, right? So they stop after cytokinesis. So the cell cycle can be defined as the sequence of events which occurs between one cell division and the next. There are three main stages as we already saw. There's interphase, mitosis and cell division. This mitosis or division of the nucleus is also called karyokinesis. So here we can see the phases of the cell cycle in a table. And these are already things that we discussed, right? So in CAP1 phase, we have the cell with the chromosomes which are not duplicated. So we have two. We have chromosome set and a paternal chromosome set. So you have 46 chromosomes. So during this phase, there is synthesis of new cell organelles and cell growth occurs, and the normal cell activity occurs during this phase. Then during S phase, DNA replicates, so it forms chromosomes with sister chromatids like this. So then in the growth 2 phase, you have division of mitochondria, and it prepares for mitosis by forming the mitotic spindles. Then you have the mitosis and finally the cytokinesis. If you talk about the interphase again, I have already mentioned this, right? So you have the genetic material which can be seen as chromatin, right? So the cell performs their normal function and DNA replication occurs. Mitosis results in two daughter cells which are identical to the parent cell. This mitosis occurs in somatic cells. Somatic cells are all cells in the body except for gametocytes. Gametocytes are sperms and ova, which we need for sexual reproduction. So skin, intestine, liver, stomach, anything, all the cells in the body are somatic cells. You have four phases: prophase, metaphase, anaphase, and telophase. So in prophase, you have the cell like this, and in this phase, you have the nucleus and the nuclear membrane. This nuclear membrane starts to degenerate, and within the nucleus, these chromosomes start to condense and appear properly as chromosomes. Right. So then you have microtubular structures called centrioles. During the prophase, these centrioles start moving towards the opposite ends of the cell. Yeah, so that's what happens during prophase. The chromosomes shorten and thicken and can be seen as the two sister chromatids. The nuclear wall degenerates, nuclei disappear, centrioles move to the opposite poles, and spindle fibers are formed. Then you have the metaphase. So you have the cell like this, and our chromosomes start to arrange in the central area. The central area is called the equator. This central area is called the equator of the cell. So on either side, we had the centrioles, and these centrioles had spindle fibers. These spindle fibers come and connect to the centromeres. So there are some fibers connecting the two centrioles. So the chromosomes arrange individually around the equator. So here you can see the metaphase. The chromosomes, that is pairs of sister chromatids, line up individually around the equator of the cell. And the spindle fibers come and attach to the centromeres from either side. Then you have the anaphase. During the anaphase, the centromeres divide. Sorry. And the two sister chromatids separate. The sister chromatids get separated and they move to the opposite poles as chromosomes. Now, after they are separated, they can be called chromosomes. So, these fibers shorten, so they are pulled to the opposite sides, and the fibers which connected the two centrioles, they lengthen. Okay. So, the sister chromatids move to the opposite sides. So here you can see the centromeres are divided and the sister chromosomes move to the opposite sides. Then you have the telophase. You have the cell like this and sorry, the cell elongates and then you have the nuclear membrane reforming, and in the nucleus you have the separated sister chromatids as chromosomes, and these start to uncoil and form chromatin, right? And these middle fibers, they also start to degenerate. So here you can see the chromatids, which is the opposite poles of the cell, and the spindle fibers degenerating, and the nuclear envelope reforming. Right? So then you have cytokinesis. Cytokinesis is division of the cell membrane. So the cell membrane bends in like this, invaginates, and finally meet each other and form two separate cells. So we started with a cell which had one maternal nucleus, one set of maternal chromosomes and one set of paternal chromosomes. So similarly, we end up with two cells, each having a set of maternal chromosome and a set of paternal chromosomes. So here you can see the microscopic images, the cell separating. So during this phase, the organelles also become evenly distributed towards the two poles. And here you can see how the cell membrane starts to invaginate, which finally results in two similar looking daughter cells. So if you look at the significance of mitosis, it has genetic stability. It maintains the same amount of genetic material in the parent cells. Right? So as we talked earlier, it is required for growth, for cell replacement and regeneration. Right? So for all this, you can replace the cells with equal looking cells. Right? So then we come to meiosis. In meiosis, we start with the diploid cells, that is a cell having a set of maternal chromosomes and a set of paternal chromosomes, and we finally end up with a set of chromosomes, only one set of chromosomes, that is so we have 46 chromosomes in the parent cell and we end up with 23 chromosomes in the daughter cell. Okay? So this is called a diploid cell. It has two sets, and this type of cell which has only one set of chromosomes is called a haploid cell. So this occurs in the formation of gametocytes that are formed during sexual reproduction. So in the formation of ova and sperms, we have meiosis. So meiosis has two parts, right? Meiosis 1 and meiosis 2. So we'll talk about this in detail later. So meiosis has a prophase 1, metaphase 1, anaphase 1, telophase 1, and prophase 2, metaphase 2, anaphase 2, and telophase 2. So during interphase 1, it's similar to the interphase of the mitosis. You have the nucleus, and in the nucleus, DNA replication occurs. So the chromosomes form two sister chromatids like this. So cell growth occurs, the organelles divide, all this occurs in interphase 1. So then prophase 2. So we take a cell, and during prophase 1, the nuclear membrane starts to degenerate and the centriole starts to move to the opposite sides. And we have the chromosomes like this. We'll take two sets. So during prophase 1 of meiosis, the homologous chromosomes come together. These two and these two come together. So the maternal chromosomes and the paternal chromosomes, which code for the same genetic material, come together and form complexes like this. They are called synaptonymal complexes, and they cross over each other. At some places, they cross over each other. Like this, they cross over. These places are, these crossing over places are called synapses, and these places called a chiasma. The plural word is chiasmata. So finally, you have a separation of these cells with exchange of genetic material. So this maternal chromosome received a part of the paternal chromosome, and the paternal chromosome received a part from the maternal chromosome, like this. So this is called crossing over or recombination. Okay, this helps to increase the genetic variation of the gametes that are made. Okay. So here you can see the prophase 1. Right? So the chromosomes shorten and thicken. The homologous chromosomes form pairs. So these pairs are called bivalents. So a pair like this is called a bivalent. So the paired chromosomes join at points called chiasma, and crossing over and recombination can occur. So similar to the prophase of the mitosis, centrioles migrate to the opposite sides, nuclear envelope is dispersed and spindle fibers form. So the prophase can be divided into several subdivisions. Leptotene, zygotene, pachytene, diplotin, and diakinesis. So, as I have already discussed, so this condensation of chromosome occurs first, which is called leptotene, and during zygotene, these synaptonymal complexes are formed, forming synapses. And during pachytene, the crossing over occurs, and during diplotin, this complex degenerates, and during diakinesis, you have the separate uh, chromosomes. It exchange genetic material, and you have the rest of the things happening, like dissolution of the nuclear membrane. Then you have metaphase 1. So during metaphase 1, the the homologous chromosomes come to the equator as pairs. So here we have the exchange of genetic material. So in mitosis metaphase, you had the chromosomes lining up individually, but during meiosis 1, during metaphase 1, these arrange as pairs. So you have centrioles on the opposite ends, and these attach to each chromosome like this. So here also you have a special thing you have to know. It is independent. So during this phase, these chromosomes can line up in the equator independent of each other. So you can see here the maternal chromosome, one of the maternal chromosomes are lined up in the upper region, and the other is lined up in the lower region. So this can also be arranged like this. The maternal two chromosomes can be on the same side, and the paternal ones on the same side. So this is two. In normal human beings, you have 23 pairs. So there's a lot of combinations which could arrange in this equator. So they can be arranged in many ways. Right? So here you have the metaphase. You have the individual chromosomes as pairs lined up in the equator. So independent assortment occurs, and the spindle fibers are attached to the centromeres of each chromosome. Okay. So then you have the anaphase. You have the cell like this, centrioles, and you have the whole chromosome without separation of the sister chromatids moving to the opposite poles. And you have the exchange. So here, these strands shorten, the strands between the centrioles lengthen, and they are separated to the opposite poles of the cell. So here you can see the whole chromosomes moving to the opposite sides. Okay. The centromeres don't divide during this phase. Then you have telophase. During telophase, you have the whole chromosomes like this. So you have the nuclear wall reforming and the centers they are degenerating, and the chromosome DNA coils separating and forming chromatin like that. You have the telophase 1. So now in each cell we have only one set from the homologous chromosome. So you have only the paternal chromosome here, only the maternal chromosome here, like that. So the 2N cell became a haploid cell. So the diploid cell became two haploid cells. So still we have two sister chromatids here. Right? So yeah, the spindle fibers degenerate, chromatids uncoil, nuclear envelope reforms, and cell division occurs. So then we have interphase 2. So interphase 2, the other things happen, right? So the cell cytoplasm growth occurs, the organelles divide, all that occurs, but DNA replication does not occur. So we ended up with chromosomes like this. So this does not divide further. They remain as they are. So then we have prophase 2. We have two cells like this, and here we have the maternal chromosome. Maternal chromosome. Maternal chromosome. This phase we have the nuclear wall degenerating and the centrioles moving to the opposite sides. So this phase is similar to the process of mitosis. So there is no complex occurrences like during prophase 1. So then you have metaphase 2. We have the two cells, the centrioles on the sides, and you have the chromosomes lined up in the equator of the cell. exchange genetic material here. And then you have the spindle fibers attaching to either side of the centromeres. So this is similar to metaphase of mitosis. The chromosomes are arranged individually at the equator of the cell. So you can see the chromosomes arranged individually and the spindle fibers attached. Then you have anaphase. You have the centrioles. Sorry. You have the chromosomes separating. So the sister chromatids separate from each other, forming chromosomes. Right. So these are pulled to the opposite ends of the cells. So you can see the centromeres divide and the sister chromatids are pulled to the opposite sides of the cell as chromosomes. Then you have telophase too. So the cells elongate. You have the so you can see we finally end up with one set of chromosomes. So cytokinesis occurs and these separate. You can see the all the four cells are different. None of the two are similar. So this is only an example with two cells. But if we had 23 pairs, there would be a wide variation among these four cells. So this during meiosis, there is a wide variation in the genetic material in the resulting daughter cells. Here you can see the spindle fibers degenerating, chromatids uncoiling, and the nuclear lock reforming. So cytokinesis finally occurs and results in 4 haploid cells. So if we talk about spermatogenesis, in the testis we have cells known as spermatogonia. So these divide and form primary spermatocytes. So spermatogonia divide by mitosis and form many primary spermatocytes. So these primary spermatocytes undergo meiosis. Meiosis 1 and form 2 haploid cells. So these are all diploid. And this is called secondary spermatocyte. and these cells undergo meiosis 2 and form 4 haploid cells. These are called spermatids. These spermatids undergo some changes and finally form sperms. So one primary spermatocytes give rise to 4 different sperms, like this. Okay. So I had it. So this is spermatogenesis. Then we have oogenesis. That is formation of a ovum or an egg. So in a female fetus, we have cells called oogonia. So in the fetus, these undergo mitosis and form primary oocytes. Okay. So this occurs before birth, before the birth of a girl, right? So when a girl is born, they have a certain number of primary oocytes, right? So all you have this number of oocytes does not change after birth. So until death, you only have this number. So these remain as they are in the ovaries. So in the ovary, you have these primary oocytes. So they remain like that until puberty. So after puberty, in each menstrual cycle, a certain number of these primary oocytes undergo maturation. That is, they start meiotic division. So they undergo meiosis 1 and form secondary oocytes. So actually, they start meiosis 1 and progress to meiosis 2 and get arrested in the metaphase 2. This is not the secondary oocyte. This is the secondary oocyte. It is arrested in metaphase 2. So you have the formation of a secondary oocyte. Cells undergo meiosis 1 and form the secondary oocyte. And one cell is called a polar body. It has very less amount of cytoplasm and organelles. Only one cell has a significant amount of cytoplasm and organelles. So this is stuck in metaphase. Right. So they enter the fallopian tubes in the uterus. You have the fallopian tubes like this and the ovary like this. So the oocyte which is stuck in metaphase 2 enters the fallopian tubes. So if a sperm enters and penetrates the secondary oocyte, it progresses to continue with meiosis 2. So again a polar body is formed and complete oval is formed. So when the complete oval is formed, it also has the paternal uh, nuclear material, genetic material within it. So it forms the fertilized egg. Okay. So here I have two diagrams. You can go through them slowly, right? So if you consider the cell cycle of the germ cell, it's not similar to mitosis. You have the ovum and the sperm. They fertilize and form the zygote. The zygote matures into a total human being, and these two adults form or again ova and sperms. So this can be considered as the cell cycle of the germ cell. Right? So then, if you talk about the significance of meiosis, it's needed for sexual reproduction. So it forms two haploid cells, and two haploid cells get together and form a diploid organism again. So, and genetic variation, you already saw how crossing over and independent assortment occurs. So that's all about the cell division and cell cycle. You can leisurely fill this chart. So, thank you.